This is an optional extra example that just shows you, again, kind of going through how the chi-squared analysis would be done. So this is an example of two different genes, and we want to do some crosses and try and find out if these are linked genes or not. And so the examples are dumpy. So these are male and female flies. You can see the normal wings extend far past the abdomen. Whereas in individuals that are homozygous mutant for dumpy, the wings they have this sort of shortened, so the shape is like that. They're much shorter, and although part of the wing extends quite far down, most of the wing it's like it's been cut. So that's what those look like. So and they're quite striking. You can see them very easily under the microscope. And then this is not a great picture, but the eyes of fruit flies are red. This is kind of almost too bright, really. It's more of a duller. I think that these are more realistic, and this is a real picture of a real fly, so you can actually see what the eyes really look like. But the normal wild type is red, but there's a huge variety of different eye color mutants in flies, and one of them is called sepia, and it produces this sort of yellowy brown color, and it's very distinctive from wild type, so it's very easy to see. And so some crosses are done. So a strain of flies, pure breeding strain of flies that have both the dumpy and the sepia mutations across to wild type. Remember that the parents are, unless you're told otherwise, they are pure breeding. And so the F1 are going to be heterozygous for both genes. Remember also that this dot, I'll say it again, I've said it a few times, it means we don't know yet what the status is of these genes. Are they on the same chromosome or are they on different chromosomes? That's what this cross or these crosses are going to tell us. So when we look at these F1, we know the F1 will have to be heterozygous because the two parents were pure breeding. And because they're showing the wild type for both traits, the wings and the eyes, then we know the mutations are recessive and the wild type is dominant. The definition of heterozygous is when you have two different alleles. The one that, the definition of dominant is that when you have two different alleles, what phenotype are you observing? And so if you're ever asked which allele is dominant, it's helpful if you mention heterozygotes when you're describing that. The F2 that we derive from breeding the males and females of this genotype, if the genes were unlinked and sorting independently, what would you expect? So you want to stop the video now and write down or think about what the expected ratio should be. That is one that you do need to just have in your memory banks. If you are asked a question about it on an exam and you have to stop and draw out a Punnett square to generate that ratio, it's going to take too much time. It's one we want you to remember. Okay, and so if you didn't get that, this would be our expected ratio. And this is a bit of a shortcut. So the plus just means wild type, and that's the dominant allele. The slash, of course, means the same as it would if we had written it, like this, where this could be either the dominant or the recessive allele. If the individuals are homozygous, they're going to show the dumpy, and homozygous the sepia, and then homozygous for both, they'll show that. And so these are our observed ratios, numbers, 730, 220, 225, and 89. This looks a lot like a 9:3 to 3:1 ratio to me. It's not perfect. If I was to round this off to 90, I would expect this to be 3 times as much, 270, and then this to be 9 times as much. So the numbers are not perfect, but we're seeing a lot of these an intermediate number of these two classes and a very small number of these ones. And that's exactly what a 9 to 3 to 3 to 1 ratio would look like. So we calculate our expected. We take the total. And this time I used numbers that were not like 1,000. But I used a number that's a bit more realistic. In real crosses, we might expect numbers like that when we're really doing the experiments. And so we take the total and then we multiply by 9/16 and we get 711. 3/16 we get 237, and 1/16 we get 79. So this number and this number are a little high, this number and this number are a little low. Okay. And so just pay attention to how we do this calculation. How do we get these numbers? And also note that if this had been 709.83 or something, I would have rounded it off to 709.8. And it's true that you can't get 0.8 of a fly, but our stats won't come out right unless we do use those. Especially in a case like this, where you might be tempted to round one up and one down, and that's going to skew your results. You've got to be super clear. Okay, so this is our null hypothesis, which is really the only one we talk about when we're doing chi-squared. The observed data are showing us this kind of ratio, and which would suggest that these genes are sorting independently, and the numbers that we're seeing are off just due to random variation. You know, that's what you want to include in your hypothesis. Okay, so here's our table. Same categories. Here's our observed and our expected. Here's our observed minus expected. Notice, you know, again, the negative numbers. I'll draw your attention to that because a good way for us to check whether we've added, you know, done our calculations correctly is that when you add all these together, you should end up with 0, and you do. 19 plus 10 is 29, minus 17 minus 12 is minus 29, so that should give us 0. Our observed minus expected squared in each case, and then divided by the expected. So this number divided by 711, this divided by 237, this divided by 237, and this divided by 79. And that gives us these numbers. And we add them all together. So for the whole experiment, this is our total chi-squared value. Okay, so you might want to think right now about what p-value is our cutoff. So what are we going to check for, and what are the degrees of freedom for this experiment? Because when you're reporting the results of your chi-squareds, you will always have to talk about the p-value, and you will always have to say what the degrees of freedom are. That's part of how you report it. Okay, so we compare these to the table values. We figure out our degrees of freedom. We look up the number and see where it lies on that table. So this is, well, I'll show you in a sec. Our calculated value actually turns out to be very, very acceptable because it's way up between 0.5 and 0.1. And that means the variation we've seen in the cross is easy to attribute to chance, and so that the data are consistent with our hypothesis. And I'll just reiterate that, like, when you look at the numbers we got, it looks a lot like a 9 to 3 to 3 to 1. So we're not surprised or shocked when we see that our chi-squared value is in the reasonable range, right? So this is where our number would lie, and it's actually much closer to this number than this number. So there's a very, very high chance that we would get numbers this far from the predicted values just based on chance, right? So when we report this, we say that we don't reject that null hypothesis. We don't reject the null hypothesis at 3 degrees of freedom. Our chi-squared value is much lower than the chi-squared value at p equals 0.05, and therefore we are going to basically accept our hypothesis. Okay, so that's a quick run through another example, but hopefully you see the steps, and when the steps are repeated a few times, it's easier to then start doing those yourself. So for the next lecture, what if those chi-squared results were not consistent with independent assortment? How would we interpret that? And if we do find that there are genes that are on the same chromosome, what kind of information can we use to figure out how far apart they are? And those are the questions we'll be looking at in the next lecture.